in maintaining blood in fluid state. Okay, and the role of the endothelial cells in trying to stop bleeding. Those are the things we discussed during our first lecture on primary hemostasis. So today we are going to look at secondary hemostasis, because we concluded in the primary hemostasis that hemostasis stopped at the point of platelet aggregation. That is where primary hemostasis stops. And then secondary hemostasis starts to convert the fibrin, the platelets that are linked together using the fibrinogen molecule, to convert that fibrinogen molecule that was used as a linking molecule between two platelets during the process of hemostasis into an insoluble fibrin, is what secondary hemostasis comes in to do. So today we're going to look at the process of secondary hemostasis. Okay. So secondary hemostasis of coagulation. We describe secondary hemostasis using two pathways, using two models, not pathways, using two models. Secondary hemostasis can be described using what we call the plasma-based model. The plasma-based model, and we can also use the cell-based model because of some discovery that scientists were able to discover. So there's an upgrade on description of the coagulation mechanism, because in the past, the two coagulation pathways, that is the intrinsic and the extrinsic pathway, both of which activate factor 10, the common pathway, okay, that led to thrombin generation. This the plasma-based coagulation system, in the sense that it is a pathway that is being represented as cascade, okay, in the sense that when one enzyme is activated, it in turn activates the other. That is using the plasma-based model, or the intrinsic extrinsic pathway, or the waterfall cascade. These terms still describe the plasma-based model. They are so named in the sense that they assume at that initial time that when an enzyme is activated, it in turn activates the other, hence the cascade. And the factor 10, factor 12 was described as the primary step in coagulation because it is found in the blood. Okay, because factor 12, when you look at the intrinsic pathway of coagulation, factor 12 is found in the blood. So that pathway of initiation of coagulation through factor 12 was named extrinsic pathway. The tissue factor pathway, because tissue factor is not present in the blood, the pathway describing factor 7 tissue factor pathway were named the extrinsic pathway of blood coagulation using the plasma-based model. So the coagulation cascade in a complex set of protease reaction involving different proteins. Okay, the final result of this reaction is to convert fibrinogen, as I was saying, that was used as a linking molecule during the process of primary hemostasis. The fibrinogen molecule was used to link two platelets together, which we called platelet aggregation, and that was at the point that primary hemostasis stopped. So in secondary hemostasis, we are saying that all that happens is to convert that fibrinogen that was used as a linking molecule in primary hemostasis, to convert it to a more stable fibrin, insoluble fibrin molecule. The final result of this reaction is to convert fibrinogen, a soluble protein, to insoluble fibrin strands, Together with platelets, the fibrin strands form a stable blood clot. This is the aim of secondary hemostasis. So to describe the plasma-based model of blood coagulation, we say that there are two pathways. I've already established to you that it has intrinsic and extrinsic pathway. The intrinsic pathway beginning with factor 12, because factor 12 is found in the blood, that pathway was named intrinsic pathway. Then because tissue factor is not found in the blood, the pathway of that arm was named extrinsic pathway. These two pathways that culminate into a common pathway, that's the factor 10 pathway. Okay, they combine together at this common pathway at this conversion of factor 10 to active factor 10 to activate prothrombin. Both two pathways meet at the common pathway. So using the extrinsic pathway to describe this plasma-based model, remember I've told you the plasma-based model was explained in the sense that as a cascade, in the sense that as an enzyme it activates the other one. And then there are two pathways involved in this activation, the intrinsic and the extrinsic pathway. So for the extrinsic pathway, we say that clotting begins. Clotting begins when coagulation factor 7, looking at the extrinsic pathway, we say that the name extrinsic pathway is just as a result because the tissue factor that is involved in that pathway is not found in the blood. Clotting usually begins when coagulation factor 7 is exposed to tissue factor and becomes activated. When factor 7 is exposed to tissue factor, it becomes activated. The factor 7, activated factor 7, then enters the pathway to cleave. Cleaving means to activate. Activate factor 9, which becomes activated factor 9, of course, and it binds with factor 9, okay, in the presence sorry for the network itch, sorry. So the tissue factor activates factor 11, which goes down to activate factor 10, okay, which goes down to factor 9, which goes down to activate factor 10, entering the common pathway. Okay, another pathway from that extrinsic pathway is the complex of tissue factor and factor 7 activating factor 10 directly in combination with factor 5, or they go to activate factor 9. Then in complex with factor 8, it activates factor 10. So the tissue factor 7 pathway can either activate factor 10 directly, then activated factor 10 in combination with factor 5 activate prothrombin, or it can go to activate factor 9 in combination with factor 8 to activate factor 10. These are the pathways. So looking at the diagram, you'll be able to describe the flow of the coagulation cascade. Okay. Okay. Then the intrinsic pathway, you can see here, factor 12 activating factor 11, which goes to activate factor 9. Okay, starting again, factor 9 in combination with factor 8 will activate factor 10. They enter into the common pathway. So both the intrinsic and the extrinsic pathway meets at this factor common pathway. Okay. So the common pathway, factor 7A can cleave. I've described that for you. I've already described that. Okay. So from the common pathway, prothrombin is being activated to thrombin. It's the thrombin that will now activate fibrinogen to form fibrin. Those fibrinogen will be activated by thrombin to form fibrin. Then the fibrin so formed will now cross link the fibrin molecule to form a more stable fibrin, being activated, cross-linking being activated by factor 13. Factor 13 also activated by thrombin. So thrombin is activating fibrinogen. There are so many functions of thrombin. It's activating fibrinogen. At the same time, it is activating factor 13 so that there will be a cross-linking of those fibrin monomers. At this stage of fibrinogen conversion to fibrin are called fibrin monomers. With the conversion of factor 13 to activated factor 13 by thrombin, these monomers of fibrin molecules are being cross-linked, making it a more stable mesh of fibrin to be able to stop the bleeding. So this is the plasma-based model. It's a cascade model describing two pathways of blood coagulation, describing blood coagulation in two pathways, the extrinsic and the intrinsic pathway. So, but we say that the secondary hemostasis can be described either using this plasma-based model, the cascade model, or using cell-based model. Okay. So why the cell-based model? Why the cell-based model? Why should they describe secondary coagulation using the cell-based model instead of the plasma-based model that we are all accustomed to? This is because some observations that they found out. Because for years, the coagulation cascade was conceptualized as having two distinct points of initiation, labeled the extrinsic and the intrinsic pathway. But over time, okay, it became clear that these two pathways are not independent. That is, they are not two parallel independent systems, meaning that intrinsic pathway is independent of extrinsic pathway. No, they found out that the two pathways overlap, that they don't function independently. Because why should they say so? Assuming we have a challenge with factor 7, we find out that the intrinsic pathway cannot compensate for that fault. Okay, it cannot compensate for that disorder or for that lack of factor in any of the pathway, because if they should be seen as something that is independent, it means that the extrinsic pathway should be able to complete the process of coagulation, or the intrinsic pathway should be able to complete the process of coagulation. But you find out that in a situation where someone lacks, take for example the hemophilias, where they lack factor 8, hemophilia A, where they lack factor 8, okay, assuming the two pathways are independent, the extrinsic pathway, factor 7 tissue factor pathway, should be able to compensate, okay, for that deficiency of the intrinsic pathway. But they found out that it is not so. Okay, so that's what led them to say that over time it became clear that the two pathways do not function in the body as parallel independent systems, so they overlap in their function. Deficiency in this pathway can affect the whole system of coagulation without the other pathway compensating for it, although they were described as intrinsic and extrinsic pathway. Okay, so that was the first observation. Okay, also the observation that the tissue factor 7 complex activates both factors, activate factors in both systems, suggests that they are linked. We saw it there. That was why I was showing you where the tissue factor was activating factor 10 directly. Okay, factor 10 that is involved in the common pathway. The tissue factor 7 complex was activating the common pathway directly, and it was also activating factor 9 that is in the intrinsic pathway. So that was another observation, that the tissue factor 7 complex activate factors in both systems, suggests that they are linked. The discovery combined with evolving understanding of the role of different cells, because in the cascade pathway, they are talking about enzyme activating this enzyme, activates another one. This enzyme gets activated, it activates another one. But they did not recognize the role of cells like the platelets play in the process of blood coagulation. So the this discovery combined with an evolving understanding of the role of different cells, in particular platelets, has led to cell-based model. So they try to describe blood coagulation, that is secondary phase of blood coagulation. That is why it's been described as occurring on the surface of cell platelets precisely. The cell-based model includes the important interaction between cells directly involved in hemostasis, okay? The tissue factor bearing cells and platelets and coagulation factors, okay? It more accurately represents the interaction between cellular activity and coagulation proteins, okay? Representing cellular activity and coagulation proteins that leads to thrombus formation and hemostasis. The cell-based model identified the membrane of tissue factor-bearing cells and platelets as the sites where activation of specific coagulation factor occur in a three-phase process. So the cell-based coagulation model uses a three-phase based model to describe secondary hemostasis. So we have the initiation, we have amplification or propagation, we have fibrin formation. So this is the overview of the cell-based model of blood coagulation, trying to describe blood coagulation, bearing in mind the surface of the cell where this coagulation process is happening. It's still the same process of factor 12 activating factor 11, activating factor 9, activating factor 10, and then also factor 7 tissue factor activating factor 10 or factor 9. It's still the same way, but described on the surface to bring to your attention that the platelet, the cells are contributing to the platelet, to the process, and that the process are not independent. So the cell-based model identified the membrane of tissue factor-bearing cells and platelets as the site where activation of specific coagulation factors occurs. This model suggests a three-phase process: initiation, propagation, and clot formation. So the initiation process, okay? Membrane-bound tissue factor, recognize that these tissue factors are membrane-bound, okay? Activate factor 7. Now we don't want to use extrinsic or intrinsic. We say initiation or propagation or fibrin formation. We don't want to use the term again, intrinsic and extrinsic terminologies. So, but in our extrinsic terminology, no, this is what was happening. Tissue factor activating factor 7, then the complex. To form the complex, the membrane bound complex activates both factor 9 and factor 10. Factor 10 converts small amounts of prothrombin to thrombin, which then activates factor 5 and factor 8. Okay, the same way of interaction we described in the extrinsic and intrinsic pathway. So we have the propagation phase of blood coagulation, secondary blood coagulation. On the surface of the activated platelets, okay, factor 11 activates free factor 9, which then forms the 10ase complex with factor 8. Okay? This factor 9, factor 8 activates factor 10, the common pathway, which in turn forms the prothrombinase complex, which you will still also see when you look at the extrinsic and the intrinsic pathway. The only difference is that they assume that these processes are occurring on the surface. This is the platelet cell. These processes are occurring on the surface of a platelet cell. Okay? Instead of drawing it as a cascade of enzyme activating each other and subsequently activating the other. It's still the same process we see in the extrinsic and the intrinsic pathway. Okay. Factor 10 is the prime component of the prothrombinase complex. You can see those reactions here, which convert large amounts of prothrombin to thrombin, described as the thrombin burst. Okay. So this complex is the prime prothrombinase complex, which converts at this stage large amounts of prothrombin are converted to thrombin, generating large amounts of thrombin. Thrombin amplifies the coagulation cascade through activation of factor 11. See, the thrombin goes back to activate the thrombin. This is prothrombin being activated, factor prothrombinase bursts, activating large molecules of thrombin. That is converting large molecules of prothrombin to thrombin. If you are following my laser pointer, okay, the thrombin that is being generated will go back into the coagulation system to activate loads of factor 11 to still also activate factor 9 and all the rest. So this is the propagation phase, the thrombin burst, at the phase where a lot of thrombin are being generated to go back into the coagulation system, activating lots of factor 9. Okay. Now the clot formation stage. The thrombin also were generated from those thrombin bursts. It goes back to activate more factor 9. A lot of things thrombin bursts. We also saw thrombin activating factor 13 to to cross-link the fibrin that is formed. So you can sit down and write out the functions of thrombin in the coagulation cascade. When you sit down to actually write the functions of thrombin in coagulation cascade, you find out that it has a lot of function to play. Okay, so in the process of clot formation, that thrombin burst that is generated at the propagation phase converts fibrinogen to fibrin monomers, first of all to monomers, which form fibrin polymers. Thrombin activates factor 13 to activated factor 13, which cross-link fibrin strands, leading to fibrin scaffolding that, along with blood vessels, form a stable clot. Thrombin further promotes clot formation through activation of platelets. You see thrombin, it activates platelets, activates factor 13, activates fibrinogen, goes back to activate factor 9. So when you sit down, write out the roles of thrombin so that you'll be able to memorize it. Okay, so the coagulation mechanism does not go on indefinitely without being regulated. Once the process of coagulation has been initiated, it has to be regulated so that the clotting will not continue. If it is allowed to continue, it may obstruct the blood vessels. So after the body coagulation mechanism have tried to arrest the bleeding, okay, there should be a regulation of the mechanism to stop further blood clotting. Okay, so we call it regulation of the coagulation pathway. It has to be regulated. Okay. So we are going to discuss three major pathways of this. We have the protein C regulatory system. We have the antithrombin tissue factor pathway inhibitor mechanism of regulating blood coagulation. Okay, so the protein C regulatory system, let me see if the diagram is here. Okay, you can see let's just look at this diagram where then we go back. Okay, you have the protein C regulatory mechanism. This is the protein C, endothelial protein C receptor pathway of regulating the blood coagulation. Okay, let's describe it first. So protein C regulatory system, it doesn't function alone. It functions with protein S as a cofactor. So protein S is the cofactor that binds and stabilizes activated protein C. Okay. It is synthesized in the liver and circulates in the plasma in two forms, free and bound form. About 40% of protein S is free, but 60% is covalently bound to complement and control proteins. Okay, bound protein S cannot participate in the protein C anticoagulation pathway. Endothelial cell protein C receptor. Okay, endothelial cell protein C receptor is a transmembrane protein that binds both protein C and activated protein C adjacent to the thrombomodulin thrombin complex and augments the action of thrombin thrombomodulin at least fivefold in activating protein C to a serine protease. So the protein C regulatory pathway needs some assistance from protein S as a cofactor, okay, to be able to function. Activated protein C dissociates from EPCR and binds its cofactor, free plasma protein S. We're just trying to describe how the combination between activated protein C and protein S occurs. That's just the meaning of that sentence. The stabilized APC protein S complex hydrolyzes and then inactivates factor 5 and factor 8. So after the formation of the activated protein C protein S complex, what this combination of APC and S, activated protein C and protein S complex does, how they regulate coagulation, is that they hydrolyze or inactivate factor 5 and factor 8. And you take your mind back the cell-based model or the cascade plasma-based model to see the positions of factor 5 and factor 8, okay, in the coagulation mechanism. Okay, so what this APC protein S complex does is to inactivate factor 5 and factor 8. That is how they regulate or stop blood coagulation, slowing or blocking thrombin generation, okay, or coagulation. That is how protein C pathway, activated protein C pathway, contributes to the process of regulating blood coagulation, okay, by the complex of APC and protein X shutting down factor 5 and factor 8. Now we have antithrombin. Okay, antithrombin is a serine protease inhibitor that binds and neutralizes the serine protease thrombin, including serine proteases, including thrombin as an example, and factors 9, 10, 11, 12, prekallikrein, and plasmin. So antithrombin has the capacity to inactivate all these factors in the coagulation pathway. Okay. AT's activity is accelerated 2,000-fold by binding to heparin and is the basis for the anticoagulant activity of pharmaceutical heparin. Okay, this is how heparin functions in the body, by binding antithrombin. AT preferentially inactivates factor 10 in vivo. Antithrombin covalently binds thrombin, forming an inactive thrombin-antithrombin complex, which is then released from the heparin molecule. So the main takeaway from here is that antithrombin blocks or slows down blood coagulation by neutralizing factors such as thrombin, factor 9, factor 10, factor 11, factor 12, prekallikrein, and plasmin. So you can see that here. You see antithrombin here. It goes to shut off thrombin, shut off factor 10 from participating in blood coagulation, shut off factor 9 from participating in blood coagulation. And when that happens, the process of blood coagulation will stop. So we also have the tissue factor pathway inhibitor. These are the three major ones we want to discuss at this level. We also have the tissue factor pathway inhibitor. Tissue factor pathway inhibitor is a Queen's type serine protease inhibitor and is the principal regulator of the tissue factor pathway. TFPI is synthesized primarily by endothelial cells and is also expressed on platelets. During the initiation of coagulation, activated factor 7 and tissue factor combine to activate factors 9 and 10. We saw that when you look at the coagulation cascade, you see the pathway from which the tissue factor factor 7 complex goes to activate factor 10 and factor 9. Then the tissue factor pathway inhibitor. See, tissue factor pathway inhibitor inhibits coagulation in a two-step process. So it's going to inhibit the process of that tissue factor pathway factor 7 complex, which we call the extrinsic pathway, using the plasma-based model of cell coagulation. First, binding and inactivating factor 10. The tissue factor pathway inhibitor, factor 10 complex, then binds to tissue factor 7, forming a quaternary complex and preventing further activation of factor 9 and factor 10. So the tissue factor pathway inhibitor inhibits the process of blood coagulation by forming a complex with factor 10, activated factor 10, preventing the activation of factor 10 by tissue factor 7 pathway. Alternatively, TFPI may bind to factor 10 in the TF factor 7 factor 10 complex and inactivate factor 10 and tissue factor 7. Tissue factor pathway inhibitor provide feedback inhibition because it is not actively engaged until coagulation is initiated and factor 10 is activated. So it can form as a sign for feedback mechanism. So you can see the diagram we use to represent the tissue factor pathway inhibitor. Okay, you can see it in complex with protein S, goes to shut off factor 10, also goes to shut off the factor 7 tissue factor pathway. So the take home here is for you to know the mechanism specifically at this your level, specifically to know the mechanism through which each of these regulatory pathways contribute to the process of blood coagulation. Okay. So we look at fibrinolysis. Fibrinolysis is the systematic accelerating hydrolysis of fibrin by bound plasmin. Two activators of fibrinolysis, plasminogen activator, TPA and UPA, are released in response to inflammation and coagulation. Fibrinolytic proteins assemble on fibrin during clotting. Okay, what's fibrinolysis come to do? Okay, the fibrin molecules or cross-linked fibrin, stable fibrin clots has been formed. Okay, the body needs a mechanism, okay, after healing has completed, to resolve what we'll call it, to maintain the endothelial cell, the patency, and maintain the smooth surface of the endothelial cells, because those fibrin clots that have been formed are not left there. If they are left there, clotting can, they can trigger clotting in vivo. So there is a mechanism by the body system to try to resolve, smoothen the surface, okay, where where fibrin has taken place. Okay, smoothing the surface, make it smooth again so that blood and cells can move inside the blood vessels without encountering any blocks. Because any hindrance, like we see in atherosclerosis, that is what in atherosclerosis, where we have a fat deposit in the blood vessels, those fat deposits trigger platelet aggregations and clumping and clotting, and at the end of the day, obstructing the vessel from allowing blood to flow. So the body has a mechanism in a normal physiologic process. After fibrin has formed in the body, there is a mechanism, fibrinolysis process, by which the fibrin strands are resolved to maintain the smoothness of the endothelium of the blood vessels. So that is what fibrinolysis has come to do. Okay, engaging plasmin, tissue plasminogen type. We also have the urokinase type. We also have PI1 plasminogen activator inhibitor become incorporated into the fibrin clot as they bind to lyse through their Krengle loops, thereby concentrating and localizing them to the fibrin clots. Okay, tissue plasminogen activation, urokinase plasminogen activator, activate fibrin-bound plasminogen, okay, several hours after thrombus formation, degrading fibrin and restoring normal blood flow during vesicular repair. This is what you want to know. You should know what is the significance of fibrinolysis in the process of blood coagulation. That is what is summarized in this slide. TPA and UPA activate fibrin-bound plasminogen, okay, several hours after thrombus formation, degrading fibrin and restoring normal blood flow. So this tissue plasminogen activator activates the plasminogen that were bound to the fibrin as it is being formed. These plasminogen activators will activate this plasminogen, forming plasmin. Okay, so when you look at this diagram, you can see the plasminogen activator. This is plasminogen. Okay, activates plasminogen to form plasmin. It is this plasmin that goes, you can see the thrombus that is being formed. It is this plasmin that goes to degrade this fibrin into monomers, generating the product we call D-diamers. And in the laboratory, we evaluate this D-dimer to know whether someone is undergoing coagulation problems. Okay, you use it as a marker of inflammation and DIC in the laboratory, because it's generated through this process of blood coagulation. You can only see the diamer. It will indicate for you that coagulation completed. There was a complete coagulation started and it completes into a generation of fibrin mesh, okay, and then degrading that fibrin is what gives us the diamer. So it helps us to monitor patients that are having some coagulopathy disorders, knowing the extent of the disorder. It helps as a marker. Inhibition of fibrinolytic system may occur either at the level of PA by specific plasminogen activator inhibitor, or at the level of plasmin, mainly by alpha-2 antiplasmin. So both process, there is control, there is check in every point of coagulation mechanism. You really have to understand this mechanism. What is the essence? What is it contributing? How is it contributing to the process? What is it trying to do in the process of blood coagulation? We've discussed the fibrinolytic system trying to maintain the patency of the blood vessel, but the fibrinolytic system also is being regulated. Okay, so we'll discuss disorders of hemostasis, or should we discuss it next time? I don't know when we'll have time to discuss it again. We'll go ahead to discuss disorders of hemostasis, please. This is a marathon lecture now. Go ahead to discuss disorders of hemostasis, okay? All right. Okay, we'll do a marathon class today. Hey, all right. So disorders of hemostasis. We are going to take the basic primary, describe it in a very primary way for you to understand it. We are not going to make it a very complex scenario at this level. So disorders of hemostasis, our outline, we are going to look at disorders that are due to deficiency or defect in the primary mechanism of hemostasis. There are disorders that are due to defects in the secondary hemostatic mechanism, and there are other disorders. Okay, so we'll see how far we go. The objective of this class, okay, is for you to be able to classify and describe clinical manifestations of disorders of hemostasis. I'll try to give you a summary how you always go about to look out for what will make you to be able to grasp and be able to describe this or disorder as fast as possible. Once you have grasped the understanding, the basic understanding, you should be able to know how to put it together in any situation. Describe the pathophysiology of coagulopathy. The pathophysiology, that is the underlying mechanism through which those disorders are manifested. Enumerate and describe the laboratory tests for evaluating hemostasis. Enumerate them. Contrast typical presentation of patients with platelet disorder versus coagulation disorder. So primary hemostasis, you have to recall, you have to remember, you have to ask yourself, what is primary hemostasis? What happens during the process of primary hemostasis? Those 4 steps, or how many steps we outline: platelet adhesion, okay, release of vessel contents, adhering to subendothelial cell. Those 4 processes and the receptors that are involved in each of them have to be in your head before you can understand any disorder, because any disorder we are discussing does not happen in the air. It happens at a specific point in that process of primary hemostasis or secondary hemostasis, and that is what you're going to, once you pick the specific aspects of the pathway the disorder is reflecting, you don't have problems. So primary hemostasis, we're trying to describe primary. This is just an overview. Okay, we have those steps we described. Say transient vasoconstriction, platelet adhering to von Willebrand factor. Okay, platelet release of ADP, thromboxane A2. Okay, recruitment and all that, and then exposure of the this receptor glycoprotein 2b3a, and the final stage of platelet aggregation. This is just an overview. Now, when you look at this diagram, we'll still discuss it. You find out that there's a deficiency we call Glanzmann thrombasthenia. Why? Because this glycoprotein 2b3a complex is deficient. That is what gives rise to the clinical presentation that's referred to as Glanzmann thrombasthenia. Then we have, these are deficiency disorders of primary hemostasis because they are affecting the process involved in primary hemostasis. That is why they were called disorders of primary hemostasis, because though the pathophysiology of the disorder affects the process involved in primary hemostasis. Now we have another one, Bernard-Soulier syndrome, as a result of defect of the glycoprotein 1b complex. You now ask yourself, this defect of glycoprotein 1b complex will hinder the adhesion. You go and remember what glycoprotein 1b does. Where is it involved in the process of primary hemostasis? It's involved in the adhesion process. And then Glanzmann thrombasthenia will affect the process of platelet aggregation, because the receptor that is defect in the disorder is glycoprotein 2b3a, which is involved in platelet aggregation. This, if you understand the fundamental of what I'm telling you, there is no disorder you look into that you will not understand. Okay. My slide is misbehaving again. I don't know what I would do now. Don't know why it started misbehaving again. Oh no. What do I do now? Shut up, what to do. Okay. I have to go back and redo my slides, and maybe we'll have to stop here. Find another time. We'll take disorders of hemostasis and laboratory investigation. Let me see when I can get time this weekend again so that we'll do it. Thank you. If you have any questions, please go ahead and ask. Thank you. I'm going to